have seen that anaerobic respiration in yeast yields alcohol, and carbon dioxide and energy. A little energy, but just enough energy to sustain the life of these yeast cells. Now, we're going to see how we can carry out an experiment to prove that actually carbon dioxide is produced during respiration of yeast. And so, we're going to start right away with the materials that we shall need. We shall need a glucose solution, 2 test tubes, a delivery tube, and lime water, oil, and yeast. We shall know how to use each of these. First and foremost, we boil the glucose solution to drive out all oxygen, because yeast does better without oxygen. Actually, it drives out all the air from the solution. After that, we get our test tube, one of the test tube, and adds oil to it, and then add glucose solution using a pipette or a dropper. And then after that, we connect it to a delivery tube, like we're going to draw through a delivery tube to another test tube containing lime water. So this is our diagram of how our apparatus should be like. So in the first place, we pour oil in this test tube, and then using a dropper or a funnel, a thistle funnel, we add our boiled glucose solution to the oil. Of course, the solution will come down and the oil will sit on top. The oil will also prevent entry of oxygen, entry of air. So this is our oil layer. And this is our glucose solution, boiled glucose solution. We also added yeast here too, and yeast, because it's actually the one that's going to feed on the solution of glucose. Now, when yeast feeds on the solution of glucose, carbon dioxide accumulates here so that it finds its way, escapes out of the oil layer to this point here, and then goes through the delivery tube. This is a delivery tube. All the way to this other test tube that contains lime water. Now, lime water is the confirmatory, is the reagent that's used to confirm the presence of carbon dioxide in science, and it's a known truth. So, the lime water, because of the carbon dioxide that has come through, so we get bubbles here, okay, that escape, but they cause the lime water to become milky. So the observation is bubbles of a gas in lime water, and the lime water turns milky. That confirms presence of carbon dioxide, and we can be sure that the carbon dioxide came from yeast. So yeast, anaerobic respiration of glucose by yeast produces carbon dioxide. We can carry out a control experiment. Boiled yeast cells cannot carry out respiration. Therefore, the glucose remains unburnt or unused. Therefore, no carbon dioxide goes to the side, and the lime water remains milky. So it will also look like that. our experiment will still remain the same arrangement, same procedure. The only difference here is we shall have boiled yeast and boiled glucose. Solution. The difference is that we shall not have bubbles here at lime water, in the lime water test tube, and therefore we can conclude that carbon dioxide was not produced, and so respiration actually does not take place in dead cells. It only takes place in living yeast cells. Our next experiment is to demonstrate that actually heat is produced during respiration in yeast. Remember we said that during respiration we produce energy. Now some of that energy is heat energy, and it's actually the heat that keeps animals warm. Now yeast too, they produce heat actually. Now we're going to see how to carry out an experiment to actually prove that. And so what shall we need? We shall need a glucose solution. Shall have a glucose solution, about 10%. The 10% the teacher can always prepare it. A yeast suspension, 2 flasks, vacuum flasks, 2 thermometers, a water bath, cooking oil, and cotton wool. So we boil about 100 cubic centimeters of glucose solution to drive out the air, of course, because yeast respires anaerobically. That's why when you add yeast to the dough, you cover it. That's why when they add yeast to maybe banana juice, they cover it so that the yeast can carry out its respiration anaerobically. It better does so when there is an air-free condition. So we boil the glucose solution to drive out air. Let it to cool. After letting it cool, pour a half of the glucose solution. We had 100 cubic centimeters. So we pour half of it into either flask, our 2 flasks, that is 50 cubic centimeters into either flask. And then after that, add small quantities of oil on top. So this is a diagram of apparatus, and in this case we have a flask. Let's call this flask A. Now in this flask A there is a glucose solution, and a yeast suspension. Both of these are our thermometers. This one too is our thermometer. We have another suspension here for glucose. This is glucose suspension. And now we have our boiled yeast. Okay, so this is our cotton wool. Something here. So we'll let this experiment stand for a day or two while we are taking keen note of any changes in the readings of the thermometer. This is our flask B. After some 24 hours or more, we take readings from the thermometers, and we keep reading at intervals of time. We notice that where there was a yeast suspension that was not boiled, there was an increase in the temperature of this mixture here, whereas where there was boiled yeast, there was no increase in temperature. That shows us that the yeast fed on the glucose to get the energy they require for life, but also heat energy was produced as a result, and that heat energy caused the change in the thermometer reading, caused the increased temperature within flask A. But since these yeast cells had been killed by boiling them, no respiration took place, and therefore there was no change in the temperature of the solution. Therefore, we can conclude by saying that yeast cells, upon respiration, produce heat, but that only takes place with living cells. Oh, cotton is used to prevent escape of heat. Okay, cotton is used in both cases to prevent escape of heat in case produced. And so that is an experiment about how to determine whether heat is produced during respiration in yeast cells.